Hello dear students, my name is Anupam Sinha and I am a biology expert at here memoneet. Today we will be discussing the solution of VPT 14. Let's start with biology part 1 section 1 and question number 101. Let's read the question first. It says that which is considered as the first genetic material, and options are DNA, RNA, either one or two, or protein. So the answer will be option number 2, that RNA is considered as the first genetic material, and the reason is it is believed that RNA played a crucial role in early life due to its ability to store genetic material and catalyze the chemical reaction, and hence it is considered as the first genetic material. Now let's move to the next question, that is question number 102. Question number 102 states that what is the duration of replication of Escherichia coli to replicate? Replication of Escherichia coli means the time taken by the bacterium to undergo a complete round of DNA replication and divide into two daughter cells. So it usually takes 20 minutes. So the duration of replication of Escherichia coli to replicate will be 20 minutes. That is option number 2 is the correct answer. Now let's move to the question number 103. Question number 103 states that consider the Michelson and Stell experiment. If the ratio of heavy intermediate light in the first generation is 0 to 0, then what will be the ratio of heavy intermediate light in the second generation? So we have to find out the ratio of heavy light and heavy intermediate and light in second generation. So in first generation we will have two intermediates only. Let's make a diagram. Let's see, this is the first generation in which the DNA has intermediate strands only, means it has N15, N14 strand, and this also has N15 and N40 strand. This is the first generation. And in the second generation, what will happen? In the second generation, first it will segregate in different like this. It will segregate like this in four strands. And then they will form the complementary strands like this. Here it is N15, N15, then N14, then N15 and N14. First these intermediates are segregated. Now they will form the complementary strand like this, like this. And here the newly formed complementary strand will be N14. Here it is N14. And here also this will be N14 and this will be N14. So here we can see that it is intermediate type, this one is light type, this one is intermediate type, and this pair is light type. Means we have two intermediate types. Sorry, we have two intermediate types and two light types. You can see this one, two are intermediate types and two are light types, whereas we don't have any heavy type. So the ratio of H is to intermediate is to light will be, we have 0 heavy type, but we have 2 intermediate type and we have 2 light type. So answer will be 0 is to 2 is to 2. Option number 2. So answer for this question will be option number 2. Now let's move to the next question, that is question number 104. It states that what is the number of base pair present in Escherichia coli. So it is a memory based question. You have to memorize the number of base pairs present in Escherichia coli is usually 4.6 into 10 to the power 6 BP, that is base pair. So answer for this question will be option number 2. Now let's move to the next question, that is question number 105. Question number 105 states that what is the function of deoxyribonucleoside triphosphate during replication? And the options are acting as substrate, provide energy for polymerization, both 1 and 2, none. Let's see the function of deoxyribonucleoside. So deoxyribonucleoside triphosphates serve dual purpose, means it acts as a substrate and they also provide energy for polymerization reaction. And here both these are mentioned here, that in option 1 it is acting as substrate, whereas in option 2 it is provide energy for polymerization? So answer for this question will be option number 3, both 1 and 2. Now let's move to the next question. That is question number 106. Question number 106 states that the Okazaki fragments formed during replication are joined by an enzyme. Means during DNA replication process, the Okazaki fragments are formed, and which enzyme joined them? So, enzyme DNA ligase join the Okazaki fragment. This is also a memory-based question, and you have to memorize that DNA ligase joins the Okazaki fragments during replication. So, answer for this question will be option number 4. Now, let's move to the next question. That is question number 107. Question number 107 states that a failure in cell division after DNA replication leads to. So options are polyploidy does not affect in any way. This cannot be answered because this affects is good sign of evolution. This is also not a good option correct. Leads to the formation of inactive enzyme. This is also not right. So the answer will be polyploidy. That is, the failure in cell division after DNA replication leads to polyploidy. What is polyploidy? Polyploidy is a chromosomal anomaly in which organism cells have more than two complete sets of chromosomes. Usually, organisms have two complete sets of chromosomes, but in case of polyploidy, the organisms have more than two sets of chromosomes. Now, let's move to the next question. That is question number 108. Question number 108 states that from the following options, which option correctly matches with the characteristics of genetic material? In options, the characteristics of genetic material is mentioned and we have to find out the correctly match. So let's discuss first the characteristics of genetic material. So genetic material should have these qualities that is it should be able to generate its replica means it should be able to replicate. It should be chemically and structurally be stable. It should provide the scope of slow changes which is known as mutation and mutation is required for evolution and it should also be able to express itself in the form of Mendelian characters. Now let's see the options. The options are able to generate its replica. So this is right. This is the characteristics of a genetic material. Second option is chemically and structurally stable. This is also correct. It should be chemically and structurally stable. And the third option is provide scope for slow changes, that is mutation. This is also a characteristics of genetic material. So the answer for this question will be option number 4, all. All these are the characteristic of a genetic material. Now let's move to the next question, that is question number 109. It states that which of the following scheme was approved as mode of DNA replication. It is a very popular question and also memory based question. The most popular scheme for DNA replication is semi-conservative method. DNA replicates through semi-conservative method, which suggests that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parallel and one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Let's assume that this is a parental DNA. When it will replicate, first it will segregate into two strands and then after segregation it will form one parental, this is parental, and one daughter or newly synthesized strand. So it will look like this. It has one parental and one newly synthesized strand. It also has one parental and one newly synthesized strand. So this scheme was known as semi-conservative method. So answer of this question will be option number 3. Now let's move to the next question, that is question number 110. It states that what is the polarity of continuously synthesized new strands of DNA? We have to find out the polarity of continuously synthesized new strands of DNA. So we know that DNA dependent DNA polymerase catalyze the polymerization only in one direction and it is usually 5 to 3n direction and consequently one strand. The template with polarity 3 to 5, the replication is continuous. That is, this is template 3 to 5 and here the synthesis is continuous. Whereas and on the other strand or on the template, the polarity with that is 5 to 3 polarity, it is discontinuous. Means synthesis is discontinuous. Here you can see the image, it is showing the discontinuous synthesis and it is showing the continuous synthesis. So the newly synthesized DNA strands are synthesized in opposite direction. We also know that the newly synthesized DNA will always synthesize in opposite direction. Means if the strand is 3 to 5, then it will synthesize strand in 5 to 3 direction. So, thus, the polarity of the continuously synthesized new strand will be 5 to 3n. That is 5 to 3n. So, our option will be option number 1. Here it is written 5n to 3n. The answer will be option number 1. Now, let's move to the next question. That is question number 111. Question number 111. Let's read the question. Question states that the definite regions in DNA from where the replication originates are known as. And the options are ORI sequences, replication sequences, high energy sequences, none. The answer for this question will be ORI sequences. That is option 1. There is a definite region in DNA where the replication originates, and this region or such regions are termed as origin of replication or ORI sequences. So, answer will be option number 1. Now, let's move to the next question, that is question number 112. It states that which type of RNA polymerase is responsible for synthesis of transfer RNA and SN, that is small nuclear RNAs. And option are RNA polymerase 3, RNA polymerase 2, RNA polymerase 1 or none. Answer for this question will be option number 1, that is RNA polymerase 3. We know that there are 3 RNA polymerase in the nucleus and there is a clear-cut division of flavor in them. And what is lever of division of flavor? That the RNA polymerase 1 transcribes rRNAs, that is ribosomal RNA. That is RNA polymerase synthesizes or transcribes rRNAs. It will synthesize rRNAs. Whereas the RNA polymerase third is responsible for the transcription of tRNA, 5S RNA and snRNA. Polymerase third will synthesize tRNA, 5S RNA as well as snRNA. It will be synthesized by polymerase third. And the polymerase 2 transcribes the precursor of messenger RNA, the heterogeneous nuclear RNA that is hnRNA. So here in the question it is asking which type of RNA polymerase is responsible for synthesis of tRNA and snRNA. And tRNA and tRNA and snRNA is synthesized by polymerase third. So our answer will be option number 1. Now let's move to the next question, that is question number 113. Question number 113 states that during which process nucleic acid is copied from one nucleic acid to another. So we know that there are two major types of nucleic acid, that is DNA and RNA, and the process by which DNA makes a copy itself, it is known as replication, and the process by which RNA is made from DNA is called as transcription. Thus, the replication and the transcription is the process by which a nucleic acid is copied from one nucleic acid to another. So answer for this question will be option number 4, that is both 1 and 2, means both replication and transcription are the processes by which one nucleic acid forms the another. Now let's move to the next question, that is question number 114. It states that which factor serves as an initiation factor to initiate the process of transcription. So you have to memorize it, that the factor which initiates the process of transcription is known as sigma factor. And the other option are row, TA box, both 1 and B. And row is known as termination factor. Termination factor because it terminates the process of transcription. So answer of this question will be option number 1. Sigma factor is known as or serves as an initiation factor to initiate the process of transcription. Now let's move to the next question, that is question number 115. Question number 115 states that transcription and translation can be coupled in. Options are unicellular eukaryotes, bacteria, or prokaryotes, multicellular eukaryotes, both 1 and B. So, transcription and translation can be coupled in bacteria, that is prokaryotes. Why? The reason is mRNA does not require any processing to become active. In bacteria, the mRNA does not require any processing to become active. And also transcription and translation takes place in the same compartment. Why? Because there is no separation of cytosol and nucleus in bacteria. And here the translation and transcription can take place simultaneously. Hence, in prokaryote, the transcription and translation can be coupled. So, answer for this question will be option 2, bacteria or prokaryotes. Now, let's move to the next question, that is question number 116. Question number 116 states that which of the following statements matches with the silent feature of genetic code? So, let's see the first we will see the silent features of genetic codes. So we know that the codon is triplet. It means that codon has three bases like U, U, U or A, U, G. It is known as codon. And 61 codons code for amino acids, whereas 3 codons do not code for any amino acids. And hence they function as stop codons. Another feature is One codon codes for one codon codes for only one amino acid. Hence, it is unambiguous specific. Some amino acids are coded by more than one codon. Hence, the code is degenerate. The codon is read in messenger RNA in a contiguous fashion. There are no punctuation. Fifth is the code is nearly universal. For example, from bacteria to humans, triple U would code for phenylalanine. Means whether it is a bacteria or human, the triple code will always be code for phenylalanine. Also, there are some exceptions. Sixth is AUG has dual function. It codes for methionine, met, and it also acts as initiator codon. Now let's check the options. The options are one codon codes for only one amino acid. So this is correct. One codon codes for one amino acid. This is correct. Second is code is generate. Yes, we have discussed here that code is generate, degenerate. The third one is code is inversal. Yes, we have also discussed that code is inversal. That is one type of codon will code same for humans or bacteria or any other organism. So answer for this question will be option number 4. That is all. Now let's move to the next question. That is question number 117. Question number 117 states that the relationship between genes and DNA are best understood by. Options are mutation studies, Punnett square, studying about genetic code, studying ORI sequences of different species. So answer will be mutation studies. The relationship between genes and DNA are best understood by mutation studies. Why? Because mutation give rise to alterations in phenotype. Because of mutation, our phenotype changes. So the relationship between genes and DNA are best understood by mutation studies. So answer for this question will be option number 1. Now let's move to the next question, that is question number 118. Let's read the question. The question what states that regulation of gene expression in eukaryotes occurs at an option are transcription level, processing level and translational level, transport of mRNA from nucleus to the cytoplasm, all of these. So answer will be all of these. The regulation of gene expression can be regulated at several levels, and these levels are the transcription level, that is at the time of formation of primary transcript. Second is at the processing level, that is regulation of splicing. Third is transport of messenger RNA from nucleus to cytoplasm. And fourth is translational level. And we all And in option, we have all these labels. So the answer for this question will be option number 4, all of these. Now let's move to the next question, that is question number 119. It states that the elucidation of lac operin was a result of close association between a geneticist and biochemist. It is also a memory-based question, and you have to memorize the names of scientists as well as their profession. Like the scientist was genetist, biochemist, biologist, or what. So the elucidation of lac operin was a result of close association between a geneticist and biochemist, and the scientists were Jacob and Monod. Here Jacob was geneticist, whereas Monod was biochemist. So answer will be Jacob and Monod. That is option number 1. Now let's move to the next question, that is question number 120. It states that DNA elements which can switch their position or call. This is also a easy and a very common question. And the answer of this question, that is DNA elements which can switch their position, are commonly or popularly known as transposons. And they are also known as jumping genes. They are also known as jumping genes. So answer for this question will be option number 4. Now let's move to the next question, that is question number 121. Question number 121 states that in DNA, when AGCT occurs, their association is as per which of the following pairs? So we know that in DNA, when AGCT occurs, then the association will take place in this way. That is, A will pairs with T and G pairs with C. That is pairing occurs between ATGC. So let's check the option. The first option is ACGT. This is wrong. AGCT. This is wrong. This is ATGC. This is right. The pairing takes place between A and T and between G and C. So answer will be option number 3, ATGC. Now let's move to the next question. That is question number 122. It says that according to every McLeod and McCarthy, genetic material is. So it is also a memory-based question. You have to learn that according to every McLeod and McCarthy, which was genetic material, whether it was DNA, protein, or let's see the another option, that is mRNA or polysaccharide. So answer will be DNA. According to every McLeod and McCarthy, genetic material is DNA. And prior to their work, the genetic material was thought to be a protein. Prior to their work, protein was considered as genetic material, but they proved that DNA is the genetic material. Now let's move to the next question, that is question number 123. It states that Hersey and Chase demonstrated that that DNA and not protein is the genetic material. Yes, Hersey and Chase conducted an experiment there, and there he proved that DNA is the genetic material and not the protein. Now the question says that with the help of radioactive isotopes, yes, we have to find out with which radioactive isotope he proved that they proved that DNA is the genetic material. So he proved that DNA is the genetic material by using radioisotopes sulfur 35 and phosphorus 32. So answer will be option number 4. He used sulfur 35 and phosphorus 32 in his experiment to prove that DNA is the genetic material and not the protein. Now let's move to the next question, that is question number 124. It states that in DNA, if 10% of guanine is present, how much thymine is present? Means in DNA, percentage of guanine is written, and we know that A is equal to T. A pairs with T and G pairs with C. And A plus G is equal to TC. And A is equal to T and G is equal to C. So if A is 10%, then T automatically will 10%. It means A and T constitute here 20%. So and out of it means that out of 100%, G plus C will be 100 minus 20, that is 80%. So in 80%, G will be 40% and C will be 40% because they are equal. So answer for this question will be the question says that how much thymine is present. We have to find out the amount of thymine. How much if 10% is guanine. Guanine is 10%. Here guanine is 10%. It means that cytosine will also be 10% and both make 20% and 80% and adenine will 40% and thymine will 40%. So thymine, how much thymine is present? So 40% thymine is present. So our answer for this question will be third. Guanine and cytosine is given here. Now let's move to the next question. That is question number 125. It states that DNA strands are. So it is well known that DNA strands are anti-parallels. It means they run in opposite directions. So answer for this question will be option number 1. Now let's move to the next question. That is question number 126. Question number 126 states that haploid content of human DNA contains. It is also a memory based question and directly from NCERT you have to memorize this. Haploid content of human DNA contains 3.3 into 10 to the power 9 base pairs. So answer of this question will be option number 4. Now let's move to the next question, that is question number 127. It states that which of the following nitrogenous bases are common for both RNA and DNA. So we know that in DNA, that is adenine, thymine, guanine and cytosine is present. Whereas in RNA, adenine and at the place of thymine, uracil is present and guanine and cytosine present. So here in DNA, RNA, adenine, guanine and cytosine. These are common, means A, G, C is common. So let's check the option. The option is C, G, A. Yes, this is the answer. C, G, A is common in both DNA and RNA. Now let's move to the next question, that is question number 128. It states that adjacent nucleotides in a polynucleotide chain are joined by, let's see the option. The options are N-glycosidic bond, phosphodiester bond, O-glycosidic bond and hydrogen bond. So answer will be phosphodiester bond, that is option number 2. Option number 2. In adjacent nucleotides in a polynucleotide chain are joined by phosphodiester bond. Now let's move to the next question, that is question number 129. Question number 129 states that sugars are attached to the pyrimides by the formation of, and options are hydrogen bond, N-glycosidic bond, phosphodiester bond, O-glycosidic bond. So answer of this question will be option 2, N-glycosidic bonds. Sugars are attached to pyrimides by the formation of N-glycosidic bond and it forms nucleoside, means when it attached to the pyrimides by N-glycosidic form, it forms a nucleoside such as adenosine, guanosine, cytidine or uridine. These are nucleosides. Now let's move to the next question, that is question number 130. It states that cytidine is so I have just explained that when sugars are attached to the pyrimides with the help of N-glycosidic bond, they are known as nucleoside. They are known as nucleoside. An example of nucleosides are cytidine, uridine, thymidine, etc. Here in the option is nucleoside, nitrogen based nucleotide, common dinucleotide in DNA, RNA. So answer will be nucleoside. Cytidine is a nucleoside. So option 1 will be the correct answer of this question. Now let's move to the next question, that is question number 131. It states that in bacteria, catalytic RNA is found in. So in bacteria, catalytic RNA is found in 50S subunit of ribosome. So answer will be option number 2. This is also a memory based question. Now let's move to the next question, that is question number 132. Here a sequence is given A, G, G, T, A, T, C, G, C, A, T is a sequence from the coding strand of gene. It is a sequence from a coding strand of gene And the question is, what will be the corresponding sequence of transcribed RNA? So basically what happens in coding strand of the sequence of coding strand of GNA is same as the transcribed mRNA. It means the sequence in both transcribed messenger RNA and coding strand of gene is the same except the thymine, because thymine is absent in RNA. In the place of thymine, the RNA has uracil, that is uracil. So let's find out the sequence. Here it is given A G G T A T C G C A T A T. So we will replace A T. So we will replace T with U. So let's replace and find out A G G T. T will replace with U. Then again A. Then again T will be replaced with U. Then C G C A and T will replace with U. So answer for this question will be option number 1. Now let's move to the next question, that is question number 133. Question number 133 states that select the correct match. Here few matches are given. We have to find out the correct match. Option 1 is ribozyme nucleic acid. So this is the correct match. Ribozyme is a catalytic RNA and RNA is a nucleic acid. So this is the correct match. So answer of this question will be option number 1. Let's see the other option as well. The second option is F2 cross recessive parent, that is dihybrid cross. Though this is wrong. When F2 is crossed with recessive parent, it is known as test cross. Now let's move to the third. It says that G. Mendel is related with transformation. This is wrong. G. Mendel is known as, that is Gregor John Mendel is known as father of genetics, and he discovered the basic principle of heredity. So he is related with heredity. Now next option is T.H. Morgan is related with transduction. This is also wrong because T.H. Morgan discovered the phenomenon of linkage in Drosophila melanogaster. So this match is also wrong. So first match is the correct. So our answer will be first option. Now let's move to the next question, that is question number 134. It states that select the correct match. Here also some matches are given and we have to select the correct match. So let's start with the option 1. Option 1 states that Alex Jeffrey related with Streptococcus pneumoniae. So this is the incorrect match because Alex Jeffrey is discovered the DNA fingerprinting technique. He is related with this. So this is wrong match. Now let's move to the other option, the option number 2. Option number 2, here it is written Alfred Hersey and Martha Chase is related with TME. This is also wrong because Hersey and Chase conducted an experiment where he proved that DNA is the genetic material and not protein. So this is wrong match. Third one is Jacob and Monod is related with lac operin. So this is right. Jacob and Monod proposed a model of gene regulation which is known as operon, yeah, or lac operon. So this is the correct match. Let's see the fourth option as well. Michelson and Stahel related with Pisum sativum. This is also wrong because Michelson and Stahel discovered the semi-conservative DNA replication in Escherichia coli and not in Pisum sativum. In fact, Pisum sativum was used in an experiment by Mendel. So answer for this question will be option number 3. Jacob and Monod discovered the lac operon. Now let's move to the next question, that is question number 135. It states that if there are 999 bases in an RNA that codes for a protein with 333 amino acids, the base at position 901 is deleted such that the length of the RNA becomes 998 bases. How many codons will be altered? So we have to find out by the deletion of one base, how many codons will be altered? So we know that when 999 bases in RNA codes protein for 33 amino acid, and if the base at 901 position is deleted, then the first 900 bases will be normal, means they will not be altered because 901 base has been deleted. So till 900 bases, the codon will be normal, and the bases after 901th base will be altered. However, due to deletion, the length of the RNA now has become 998 bases. Hence, 98 bases will be altered, means 900 998. Initially it was 998, but due to deletion it has become 998. Until 900 nothing has changed, so 98 bases will change. And we know that 3 bases stand for 1 codon, so 98 bases will stand for 33, means 32 point something codon, but we can't take it as this, so we will take an approximate value, that is 33 codons. So 33 codons will be altered due to deletion of one base at 901 position. So our answer of this question will be 33 codon, that is option number 3. Hope the answer is clear to you. Now let's move to the next question, that is question number 136. It states that DNA replication in bacteria occurs. So usually DNA replication in eukaryotes occurs in S phase. But in bacteria, S phase is not well marked. Hence, DNA replication in bacteria generally occurs prior to the fission. So answer of this question will be option number 3, that is prior to fission. Now let's move to the next question, that is question number 137, and it states that spliceosomes are not found in cells. So what are spliceosomes? Spliceosomes are large ribonucleoprotein complexes, and they are generally found in eukaryotic cells such as in plants, animals, fungi, etc. And they play a crucial role in splicing of introns, and that's why the name is spliceosomes. So they will found in eukaryotic cells and not in prokaryotic cells. Let's see the option. The options are plants. This is eukaryotic, fungi, animals. So all these three are eukaryotics. So a spliceosome will be found here, but bacteria is a prokaryotic. So spliceosome will not found in bacteria. So answer for this question will be option number 4. Now let's move to the next question, that is question number 138. It is a certain and reason type question. We have to find out on the basis of these four options if A and R are true and R is correct explanation of A. Here A represents assertion and R represents reason. Next option is both A and R are true, but R is not the correct explanation of A. A is true, R is false. Both A and R are false. So let's read the assertion first. Assertion says that operator gene is functional when it is not blocked by a repressor. So it is correct. Operation gene is generally functional when the repressor does not block it. Next reason is the regulator gene produces active protein which act as the operant system in Escherichia coli. This reason is also correct. Regulator gene actually produces a active protein, and that protein is known as repressor protein, which acts on the operon system in Escherichia coli. So when operator gene is functional, when it is, so the reason assertion is operator gene is functional when it is not blocked by a repressor, and reason explaining it that yes, because when the repressor, that is an active gene, is produced, it acts on operon system. So both A and R are true, and R is the correct explanation of A. So option number first will be our answer. Now let's move to the next question. The next question is also a assertion reason type question, and it states that a peptidal transferase site is contributed by a larger subunit of ribosome. So this is assertion, and assertion is true. The large ribosomal subunit contains the site of catalysis, and it is known as the peptidal transferase center, and it is responsible for making peptide bonds during the protein elongation and for the hydrolysis of peptide tRNA during the termination of protein synthesis. And reason is the enzyme peptide transferase is, this assertion is true. And reason is the enzyme peptide transferase is contributed by both 23S and 16S ribosomal units. So reason is wrong. because the enzyme peptidal transferase is contributed by 16S ribosomal unit but not by 23S ribosomal unit. Instead, it is contributed by 30S ribosomal unit. So here assertion is true but reason is false. So option number 3 will be our answer, where in this option it is A is true and R is false. Now let's move to the next question, that is question number 140. Question number 140 is also assertion and reason type question. Now let's read the both statements. Assertion says that cDNA libraries are important to scientists in human genomics. So this is right. cDNA, that is cDNA refers to the complementary DNA and it is synthesized from messenger RNA. The reason is cDNA is a synthetic type of DNA generated from messenger RNA. So it is also true. It is also true that cDNA is a synthetic type of DNA and it is generated from messenger RNA, but it does not explain the assertion that why cDNA libraries are important to scientists in genomics. So answer for this question will be option number 2. Both A and R are true. That is, both assertion and reason are true, but R is not the correct explanation for A. So option number 2 will be our answer. Now let's move to the next question, that is question number 141. It states that it is also assertion reason type question, and assertion is catalytic functions were assigned to RNA molecules during evolution. So it is right. During evolution, RNA used to act as a genetic material as well as a catalyst. And the reason was there are some important biochemical reaction in living system that are catalyzed by RNA catalyst and not by the protein enzymes. And the reason here written, the rate of mutation is quite faster in RNA. This is also true. A is true and R is also true. The rate of mutation is faster in RNA because they catalyzes the reaction, and being a catalyst, they were very reactive and hence were unstable. So mutation takes place easily there. So our answer for this question will be both assertion and reason are true. Both assertion and reason are true, but R is not the correct explanation for A. That is option number 2. Here the reason is not explaining catalytic functions were assigned to RNA molecules during why catalytic function was assigned to RNA molecule during evolution. Now let's move to the next question, that is question number 142. Question number 142 is also a assertion reason type question, and it states that polypeptide sequences are dictated by DNA and represented by messenger RNA. So this is true. Polypeptide sequences are dictated by DNA and are represented by RNA. And reason is sequence of amino acids in a polypeptide can be predicted by tRNA. So sequence of amino acids on polypeptide cannot be predicted by RNA. Cannot be predicted by tRNA. Hence the reason is false. So our answer will be A is true but R is false, means option number 3. Now let's move to the next question. Next question is a diagram based question. Here a diagram is given and we have to find out the A, B and C. Here this diagram shows that DNA is forming DNA. So this process is known as replication. So A will be replication. Here from DNA, messenger RNA is synthesizing. So this process is known as transcription. And messenger RNA is synthesizing protein, and this process is known as translation. So A will be replication. Let's check out the options now. A, transcription. So this option is wrong. This matching is wrong. So this could not be our answer. In option number 2, A is G phase. This is totally wrong. Option number 3, A is replication. Yes, this is true. A is replication. B is transcription. This is also right. B is transcription, and C is translation. This is also right. So answer for this question will be option number 3. The process A is replication process, B is transcription process, and C is translation process. Now let's move to the next question, that is question number 145. Here it is written, or this question states that in the given figure identify A and B and also identify the process. It is also a diagram based question. Now let's see the diagram. This diagram here it is given. So this, what is this? This diagram is diagram of termination process. Termination. Why? Because you can see that after this no strand cell is forming. Means the process is terminating here, and this is a DNA and this is a RNA. Means it is a process of transcription, and in transcription the stage is termination. So we can say that process is transcription and the stage is termination, or it is the termination process. And in termination process rho factor acts and RNA polymerase take part. So this will be RNA polymerase. So here uh, the process will be termination. So let's see the option. The option is first A is RNA polymerase. Yes, this is right. B is rho factor. Yes, this is right. But the process is initiation. So this is not an initiation process. This is a termination process. Let's see the two options. The options two is A is RNA polymerase. Yes, this is right. B is rho factor. This is also right. And C is process is termination. So this is also right. So answer for this question will be option two. Now let's move to the next question. Next question is also a diagram based question. Here a diagram is given. In fact, here it is written that label parts of nucleosome. This is a diagram of nucleosome and we have to label the parts. So you have to memorize the parts of nucleosome. So here this is DNA, and that is B is H1 histones. C is histone octamer. This is C is histone octamer. And what is this? This is the core of histone molecule. This is the core of histone molecules. So let's now check the option. Here in option 1 it is written A is RNA. So this is wrong. So this option could not be our answer. In option B, A represents DNA. This is right. B represents H2 histone. This is wrong because B represents H1 histone. So this option could not be our answer. In third option, A represents 2. This is correct. B represents H2 histone. It is also wrong. B represents H1 histone. Now let's see the fourth option. Here A represents DNA. This is right. A represents DNA. B represents H1 histone. This is correct. B represents histone protein here. This is histone protein. C represents histone octamer. This is also right. This is histone octamer. And D represents core of histone molecule. This is also right. This D is the core of histone molecule. So the answer of this question will be option number 4. Now let's move to the section B of this test and read the question number 146. It says that which stage of Hershey and Chase experiment is shown in this figure. It is also a diagram based question where the Hershey and Chase experiment figure is given. Let's see what image is given here. So this diagram is given here. So you have to memorize the Hershey and Chase experiment diagram, and you also have to memorize the each stage. That this is a radioactive labeled DNA. This stage is known as infection stage, and this stage is known as blending stage, and this stage is known as centrifugation. Now let's match this image with this. So here in the question this image is given. So answer for this question will be option number 2, blending. You have to memorize the Hershey and Chase experiment images and each stage as well. Now let's move to the next question. Question number 147. It states that if a DNA percentage of thymine is 20%, then what will be the percentage of guanine? Means in a DNA, the percentage of thymine is 20%. Then we have to find out the percentage of guanine. So we know that A pairs with T and G pairs with C, and the amount if A is equal to T, here T is 20%, then automatically A will also 20%. So both will constitute 40%. So left after mining 100, after subtracting 100, 40 from 100, we will get 60%. Means the remaining is 60%. And we know that G pairs with C and G is equal to C as well. So we will divide this 60% into G and C as 30%, 30%. So the answer for this question will be option 3, 30%. Now let's move to the next question, that is question number 148. Question number 148 states that gene regulation governing lactose operon of E. coli that involves the lac first gene product is. Here it is talking about gene regulation that is governing lactose operon of E. coli, and four options are given. Let's read the option. The first option is feedback inhibition because excess of galactosidase can switch off transcription. Second option is positive and inducible because it can be induced by lactose. The third option is negative and inducible because repression protein prevents transcription. And the fourth is negative and repressible because repression protein prevents transcription. So we know that lac operon under the control of repression is a negative regulation. It is a negative regulation, and the nature of the operon is inducible. The nature is inducible because depression protein prevents transcription. So answer of this question will be option number 3. Now let's move to the next question, that is question number 149. Question number 149 states that which one of the following is wrongly matched? Here matches are given and this time we have to find out the wrongly matched. So let's see. The first is it is written transcription, writing information from DNA to RNA or DNA to tRNA. So this is the wrong because transcription is writing information from DNA to messenger RNA. So this should be our answer. We have to find the wrongly matched. Now let's see the other option as well. Second option is translation, is using information in mRNA to make protein. This is correct. The process of synthesis of proteins from mRNA is known as translation. The third is repressor proteins binds to an operator to stop enzyme synthesis. This is also right. The function of repressor protein is to bind an operator to stop the enzyme synthesis. And the fourth option is operon, structural genes, operator, and promoter. This is also right. In operon, we have different genes such as structural genes, operator, and promoter genes. And we also have regulator gene. We also have regulator gene. Here it is not mentioned, but this matches although correct. Operon have structural genes, operator, and promoter. So answer for this question will be option number 1. Now let's move to the last question of this session, that is question number 150. It states that if one strand of DNA has the nitrogenous base sequence as A, T, C, T, G, what would be the complementary RNA strand sequence? So here in DNA it is the nitrogenous base sequences are A, T, C, T, and G. So we know that in RNA, at the place of T, we have U, that is uracil. So what we will do? We will do the A pair with T, but in case of RNA, here it is DNA, basis of DNA, and we will make the basis for RNA. So A will pair with U, T will pair with what? A, because A pairs with T. G will pair with, sorry, C will pair with G, and T will pair with again A, and G will pair with C. So we will have U, A, G, A, C. Now check out the options. The first option A, C, T, G. This is wrong. Second option is A, T, C, G, U. This is also wrong. Third option is T, T, A, G, U. This is also wrong. And fourth option is U, A. That is U, A, G G A C A C. So answer for this question will be option number 4. Hope you like the solution and solution is clear to you. Thank you.